A very good morning, friends. Today we'll discuss about an important topic, that is fatty acid synthesis. This is a somewhat complex topic in biochemistry. Now we'll discuss this topic under the heading of, the first we will discuss the site of this pathway. Then we'll discuss about the transport of acetyl coenzyme A, then requirements for the pathway, then activation of acetyl coenzyme A, and the pathway proper, in which we'll discuss about the regulation of the pathway. So in the last video lecture we already deal with the fatty acid synthesis complex. So today we'll start with the fatty acid synthesis proper. So site, it is de novo synthesis of fatty acid. It occurs in the liver, kidney, lung, lactating mammary gland, adipose tissue. De novo synthesis of fatty acid, it typically occurs in the cytosolic fraction of the cell. It operates in the cytosol. Now, in the transport, the basic requirement or basic precursor molecule for synthesis of fatty acid is the acetyl coenzyme A. This acetyl coenzyme A, it is generated from the pyruvate by the pyruvate dehydrogenase complex in the mitochondria, and it is also synthesized from the fatty acid oxidation or beta oxidation of fatty acid. So it is present in the mitochondria, and this acetyl coenzyme A is not able to transport across the mitochondrial membrane, so it combines with the oxaloacetate in the mitochondria and get converted into a citrate. Then citrate crosses the mitochondrial membrane, and in cytosol, with the help of ATP citrate lyase, this citrate is converted into acetyl coenzyme A plus oxaloacetate. This is how acetyl coenzyme A transport from mitochondria to the cytosol, and this fatty acid synthesis occurs in the cytosol. Now requirement, the basic requirement is the acetyl coenzyme A first, followed by the NADPH, which provides the reducing equivalent, and ATP. So this is the basic requirement for fatty acid synthesis: acetyl coenzyme A, NADPH, and ATP. So this is a basic requirement for fatty acid synthesis. Now coming to the activation of acetyl coenzyme A, and this is a regulatory step in the fatty acid synthesis. In this step, in the activation of acetyl coenzyme A, important enzyme is acetyl coenzyme A carboxylase. This is a rate limiting and regulatory step in the fatty acid synthesis. Through this step, this acetyl coenzyme A, it is converted into malonyl coenzyme A. As this is a carboxylation reaction, it requires A, B, and C. So it requires ATP CO2 is required and it is a carboxylation reaction. It requires biotin and it requires carboxylase, that is acetyl coenzyme A carboxylase. So this is a rate limiting step and this is regulated by dephosphorylation and phosphorylation and also by the hormones, that is insulin and glucagon. So this is the first step in the fatty acid synthesis, that is activation of acetyl coenzyme A into malonyl coenzyme A. Now coming to the pathway proper. For remembering the pathway, you can remember this mnemonic, that is ATM transaction kit started refund of ATM transaction kit started refund of delivered in cash rupees through ACP. This mnemonic is hard to remember, but once you remember this mnemonic, you can easily write the fatty acid synthesis process. So ATM transaction kit started refund of delivered in cash rupees through ACP. So this AT is for the acetyl transacylase, this MT for malonyl transacylase, this KS it is for ketoacyl synthase, then R for ketoacyl reductase, D for dehydratase, ER for the enoyl reductase, this is thioesterase and ACP. So these are the sequence of reactions which occurs during the fatty acid synthesis. In the first step, what happens? There are two sulfhydryl groups in the functional unit of fatty acid synthase. One is attached to the enzyme. It is cysteine sulfhydryl groups, and another is attached to the ACP through 4-phosphopentatene, that is the another sulfhydryl group. So these are the two attachment site in the functional subunit of fatty acid synthase complex. Now first step is the acetyl transacylase. So this acetyl transacylase that causes the conversion of this acetyl coenzyme A or condensation of acetyl coenzyme A plus enzyme cysteine sulfhydryl group. So this is the sulfhydryl group of the enzyme. This acetyl transacylase catalyzes the transfer of acetyl group to the cysteine of this enzyme. So it leads to the production of acetyl enzyme. It leads to the production of acetyl enzyme, and there is release of coenzyme SH form. So this is the first step. In second step, this malonyl coenzyme A, it reacts with the ACP and it gives malonyl ACP. That is through the malonyl transacylase. So this is the second step in the reaction. So in the first step, there is transfer of this acetyl unit to the cysteine of this enzyme, results in the formation of acetyl enzyme. plus coenzyme ASH, and malonyl get transferred to the ACP and leads to the formation of malonyl ACP and coenzyme ASH. So these are the two steps catalyzed by the acetyl transacylase and malonyl transacylase. Now in the third reaction, there is condensation of this acetyl enzyme and malonyl ACP occurs in presence of the third, that is ketoacyl synthase, that is ketoacyl synthase, lead to the production of beta ketoacyl ACP. In this reaction, there is liberation of CO2 occurs and there is release of coenzyme A. So this is the third step in the reaction. Coming to the fourth, in which beta ketoacyl ACP by the ketoacyl reductase, and reductase reaction requires NADPH. So in this reaction, NADPH is required, which is converted into NADP, and there is formation of beta hydroxy acyl ACP. In the next reaction, which is catalyzed by dehydratase, there is removal of water molecule and there is formation of double bond occurs, and this compound is converted into enoyl ACP. Now enoyl reductase, the next one is the enoyl reductase, act on this This reaction again requires NADPH. This NADPH which is required for this pathway, it is produced 50 to 60% cases it is produced by the HMP shunt, and it is also produced by the malic enzyme which causes the conversion of malate to pyruvate. So there are two sources of NADPH here, HMP shunt and malic enzyme. This enoyl ACP by enoyl reductase it is converted into butyryl ACP. So this is the third step, this is the fourth, this is the fifth, and this is the sixth step. So in this way, this third, fourth, fifth and sixth step repeated. There are repetition of 6 cycles occurs, and it lead to the through the multiple steps it leads to the synthesis of palmitate. Through the repetition of 6 cycles, total 7 cycles are there. With this condensation of this acetyl and malonyl, there is formation of ketoacyl ACP which is converted into beta hydroxyacyl ACP which is converted into enoyl ACP which is converted into butyryl ACP. Then there is transfer of this butyryl unit to the enzyme part of that fatty acid synthase complex, and then through the repetition of 6 cycles there is synthesis of palmitate. Now coming to the regulation, this pathway is regulated by 2 important hormones. It is regulated by 2 important states. 2 important hormones are required for the regulation. 2 states are required, that is phosphorylated and dephosphorylated state. Then the long-term regulation is by regulation of gene expression. Regulation of gene expression. It is the long-term control for the fatty acid synthesis. An important enzyme for regulation is the acetyl coenzyme A carboxylase. This enzyme, it is active in dephosphorylated state. So the insulin and thyroxine they favor the dephosphorylated state formation, hence activate this acetyl-CoA carboxylase. While glucagon and glucocorticoid, they favor the phosphorylated state formation, that glucagon and glucocorticoids, they favor the phosphorylated state formation and inactivates this enzyme. Again, there is allosteric activation. This acetyl-CoA carboxylase, it is allosterically activated by the citrate. So citrate, it is an important allosteric activator of this acetyl coenzyme A carboxy. All about the fatty acid synthesis. Thank you. Keep watching.